It's a real pleasure to be here. It's actually the first time I've been to Waterloo. I've been to Guelph once before, and this is the first time I've made that short journey, so it's a particular pleasure to be here. What I want to talk about today, this is I'm going to a fairly loosely connected series of observations about nanotechnology in general. I'll mention some of the technical experimental work from my lab, but in fact that will be quite a minor part. They're just put in as a few little observations. If anybody's interested in the technical details of those experimental things, perhaps we can talk about that afterwards. But I'm really just going to put them in to illustrate some points I want to make more generally about nanotechnology, and in particular, as the title perhaps suggests, what I really want to talk about is the relationship. What is the relationship between biology and nanotechnology? What are the lessons that we ought to learn from biology? And I think there are kind of right lessons and wrong lessons. So I'm going to start with this very famous picture that I'm sure you're all familiar with. This classic picture from Don Eigler's group in 1990, which I think, if anything, really established the idea of nanotechnology as a separate and exciting, powerful discipline. I think it was this image that did it, because what way could more graphically illustrate the fact that one can really not just look at single atoms, but one can manipulate single atoms. So it's a picture of great promise, but it's also a picture which leaves open the question, okay, this is truly cool, but beyond making nanoscale logos, what actually can one do with this? Where do you take this? So what? What does this tell us? Well, the nanotechnology, of course, is defined not by any single type of technology. It's rather a different thing. You know, people talk about nanotechnology compared to, nuclear engineering or genetic modification as technologies, but nanotechnology is really a bit different from that, because it's not really defined as a single thing. It's only defined by a length scale, and you're all familiar with what this length scale is. It's this length scale that's defined somewhat loosely at the lower end by the size of atoms and molecules. People, of course, talk about the upper limit of the length. Where does nanotechnology stop? People say sometimes 100 nanometers. I remember once interviewing somebody for a faculty position in Sheffield who was very interested in nanotechnology, actually a very good scanning probe microscopist as it happened, and I said to him, well, what do you think defines nanotechnology? And he said, oh, it's anything less than 100 nanometers. And I said, oh, why 100 nanometers? And he said, because President Clinton said so, which is as good an answer as one can ever get. And President Clinton, of course, being notorious for his accuracy with the use of language, I think that's probably quite a good statement. So it's really, what defines it is a length scale, but from the point of view of a physicist, I suppose, what we should be asking is, what's the implication of that, the fact that we are working on that different length scale? What should it make us think about the way that we do engineering? Because nanotechnology is supposed to be about making devices, making things that do interesting things. So what sort of lessons should we learn about the engineering rules? Of course, going back further in history than Don Eigler, if we ask the question, who historically is associated with the invention of nanotechnology, of course, many of you, I'm sure, will give the answer that it was Richard Feynman in this very famous lecture, Plenty of Room at the Bottom, in 1959. Of course, one of the kind of many strange twists that my career has taken in the last few years, I've been talking quite a lot to kind of historians of science, cultural studies people, philosophers of science, who are interested in nanotechnology as a phenomenon. And of course, one of the interesting things that one finds from this is, of course, this canonization of Richard Feynman as the founder of nanotechnology is entirely retrospective. You do a citation analysis on this paper, and nobody noticed it. Nobody took any notice of it whatsoever for at least 20 years. So although it's convenient for people to associate, and you can see why people have done this, because it was quite a far-sighted lecture, but more importantly, Richard Feynman has this iconic status, and so it's natural to associate him with this discipline. And in some ways, I suppose, also, it gets around a kind of problem of origins that this field has got, that its origins are more complicated than people might think. And of course, the other big figure in the origins of nanotechnology that's much more problematic and controversial is, of course, Eric Drexler. So Eric Drexler in 1986 wrote this book, Engines of Creation, which defined a particular vision of nanotechnology that's been hugely influential. Hugely influential, but of course very controversial. So it's influential in that it's his vision that is, if you like, informed nanotechnology as it appears in popular culture, you know, in video games, in television programs, in films, all that sort of thing, really comes from this particular vision of nanotechnology. But on the other hand, it's a vision that's very controversial with nanoscientists, and famously, you know, Richard Smalley had a famous falling out with Drexler. And I guess what, you know, to some extent one can say that Drexler is almost the name that can't be mentioned in polite company when it comes to nanotechnology. But partly what I want to do in this talk is actually re-examine what some of his ideas were. And I guess my position, just to anticipate things, is that actually I think Drexler was actually wrong about many things, but he was actually rather interestingly wrong. And it's actually quite instructive to ask ourselves, you know, why is it that he is in fact wrong about these things. Just to mention, of course, what the Drexler plan was. The Drexler plan was to take this idea of atomic precision machines, so taking this idea of being able to manipulate atoms individually. And then he had this really quite Fordian vision, if you like, of being able to kind of place atoms and molecules in any kind of arrangement. And I suppose the central key idea here is this idea of software control of matter, reducing matter to the status of software. So, you know, the argument is, just as, you know, we're all used now to the idea that music can be reduced to ones and zeros. Every MP3 file is the arrangement of ones and zeros that allows us to construct any arbitrary waveform that can be any piece of music. We're used to the idea of images, still and moving images, being reduced to software. So in a sense, this is this powerful and very seductive idea of being able to reduce matter to the status of software, because you know, we know matter's made out of a finite palette of atoms and molecules, and we should be able to specify their positions with digital accuracy. And of course, the point about the Drexler vision was taking this idea and taking it to the extreme limit. And in the extreme limit, one can say, well, you can make anything you like. So in this limit, you get this revolution of goods coming for free, strong artificial intelligence, because you can make anything. I mean, and it's associated with various reductionist views about consciousness as well. This idea of being able to kind of manipulate cells on a cell-by-cell basis, and hence, you know, ultimately even immortality as the result of this. And of course, such a powerful vision's got a downside as well. And you know, it's been a fertile field for science fiction. Say science fiction, of course, it's really enters into all kinds of popular culture, and you know, my regular watching of Scooby-Doo on Saturday mornings was livened up the other day by the discovery that in the new incarnation of Scooby-Doo, Scooby snacks actually contain nanobots which are used for canine enhancement. So it was I thought a very incisive contribution to the canine enhancement debate, in fact. I was only watching it because of my daughter, of course, I have to stress that. And you know, there famously books like Prey, which take this idea of nanobots being out of control. And of course, one has this wonderful and seductive idea of grey goo consuming the entire Earth. So of course, you know, with these very powerful ideas around, it's not actually surprising that this idea of nanotechnology has not been universally welcomed. And actually, we're seeing quite an interesting alliance, if you like. You know, from the political science point of view, it's quite interesting to see who's actually opposing nanotechnology. In the UK, the subject rose to prominence thanks to the intervention of this prominent scientific commentator, Prince Charles. Prince Charles, in 2003, famously wrote a number of articles, or had written numbers of articles about this terrible new technology. And so, you know, Prince Charles Prince Charles is probably more political than he ought to be, but his political stance is essentially rather romantic conservatism. He's very keen on organic food and nice architecture and that kind of thing. So from his point of view, you could see the problem. Meanwhile, across the Atlantic, other protesters took a kind of slightly more graphic approach to protesting. Of course, they're well-read protesters who'd read their Richard Feynman, so they've all got plenty of room on their bottoms for... yeah, okay. So anyway, I guess the important point about this is that it's we've seen a very kind of deliberate attempt to frame this debate about nanotechnology in the same set of ideas that genetic modification produced. So the agricultural biotechnology is very successfully opposed by environmental groups by appealing to this idea that somehow it's wrong to meddle with the basic structure of living things. And so the argument, the framing goes, well, if it's wrong to meddle with the structure of life, now we're really meddling with the basic structure of matter itself. So I mention these things because I think it's important to be aware of the kind of cultural subcurrents that are surrounding the debates about nanotechnology. There won't be a nanotechnology if there aren't people prepared to buy the products that it produces. And so I think anybody who's interested in getting the most out of the technology has a real interest in understanding this and making sure that this debate goes well. In the UK, Prince Charles's intervention had quite a good result in that it made the government commission a report. And the government commissioned a report from the Royal Society and the Royal Academy of Engineering. That came out in 2004. It's a very good report, actually, and I think everybody who's interested in nanotechnology actually ought to take a look at that. But it made this point that in terms of nanotechnology, one ought to engage the public, you ought to have a constructive and proactive debate, really before things got polarised. I mean, the argument was when it came to agricultural biotechnology, by the time people started having a public debate about it, it was too late because, entrenched positions had been taken, no one was going to change each other's minds, and the whole thing just descended into a kind of ultimately fruitless slanging match. So that actually is, the UK government did actually pretty much accept this recommendation, and so we've seen a number of activities in the UK, a number of kind of public engagement things, citizens' juries, consensus conference type things. So, here's just an example. It's just a bunch of people sitting around a table constructively discussing, in this case, this was an inquiry about the use of nanotechnology for environmental remediation. So it was an event sponsored by the Environment Agency, involved a think tank called Demos, had some government funding. And the government also set up a group called the Nanotechnology Engagement Group, just to kind of keep an eye on all these things, and I chaired this group. And the final report really, I think, was quite instructive in telling us, actually, there isn't a public backlash against nanotechnology, and to be honest, it doesn't really look like there's one in prospect. When it comes to it, when you have this discussion about the benefits and risks of nanotechnology in a kind of low-key and adult kind of way, you can find that people are really very excited about the prospects of nanotechnology. Things like clean energy and the environment have a huge resonance at the moment in the UK, and I can't imagine things are different here. Healthcare and medical applications always get a kind of a big, they always get enthusiasm. And also, people are very concerned about jobs and economic growth, and people are very willing to be convinced that new technologies like nanotechnology can lead to new opportunities for jobs and economic prosperity. But people are concerned. There are legitimate concerns about safety and toxicity that have been well rehearsed. And there's a kind of more general concern about, which is a general thing not specific to nanotechnology, that in the world that we live in, it does seem, particularly in a kind of a very heavily globalized world, it's very difficult to see who actually does control and regulate technology. No governments make a decision to introduce a technology. They just appear through the free market and through the operation of international companies that really operate quite across borders. And that is something that I think does concern people. Let's move on to what actually nanotechnology is going to look like. And I want to come back to this idea about, these Drexlerian ideas, if you like, about what nanotechnology is like. And I want to kind of cast our mind back to a reading of the history of technology that I think has been quite influential in the way people think about the subject, but ultimately actually rather misleading. Now, it's very common to think about the history of technology in terms of power. You learn about the Industrial Revolution, you learn about James Watt making better steam engines, and you learn about increasingly applying power. But there's another kind of narrative to the history of technology, which I think is less well known, but equally interesting, if not more so. And that's the history of technology as being about the history of increasing precision and increasing miniaturization. So I live in Derbyshire in England, central England, and it's an old lead mining area. So round my houses there are all these kind of old hillocks which result from medieval lead mining. And if one had gone out in about the mid 16th century, say in 1550 or so, this is the kind of thing you'd have found in the hills around my house as pretty much the state of the art of engineering at that time. So this is an illustration from a medieval mining manual from Agricola's book on mining, and it's a rag and chain pump. So you can see it pumps water out of a mine by having a continuous chain of rags that just soak the water up and are squeezed out as they come down. And so you can see the power source, it's human power. There's actually people in a treadmill that are actually making it work. So on the one hand, I'd say, this looks very crude. Everything's made out of big chunks of wood that have been put together by rather crude techniques. Adzes have been used to shape the wood. So everything's on a large scale, it's big, it's crude. But it is, it's recognisably mechanical engineering. It's recognisably, it's cogs and gears and pulleys and components that we very much recognise now. And so we can think in some ways of the history of engineering going from there through to the Industrial Revolution as being taking those kinds of mechanical engineering works and making them on a finer and finer scale. And indeed, Derbyshire, again, it's known as the cradle of the Industrial Revolution because Arkwright built his mill there as one of the first factories for making cotton in the late 18th century. And again, the people who actually made the machinery were millwrights. They were people whose trade was making windmills and water mills. So this kind of thing. But they got better at it, and they got better at making things more precisely. We then saw development of techniques for metalworking. So people like Joseph Whitworth, famous for introducing the interchangeable screw gauge. They just developed new techniques for machining things. So by the mid-19th century, people were able to machine things with pretty much, if you went to your machine shop in this university and without asking them, to work very hard, the kind of precision that they can achieve without trying very hard now with the standard machinery is pretty much what people could do in the mid-19th century, working very hard at it. So, already they were able to get precisions, less than 1,000th of an inch, so, less on the scale of tens of microns. And so, the Babbage difference engine is famous as an example of something that was just on the cusp of possibility at that time. But these developments, although they're kind of unspectacular, were very important. So, for example, I mentioned James Watt as being, we all know about James Watt and his invention of the separate condenser as being something that led to the possibility of getting much more power out of steam. That was only possible because his collaborator Matthew Boulton had new techniques for metalworking that allowed him to make the cylinders, to bore cylinders with a diameter that was well enough controlled to keep the high pressures that you needed for those steam engines. And indeed the whole factory system, this whole idea of making interchangeable parts really only came about because of precision machining. So, the classic example being, making guns. Traditionally, a gunsmith would sit there with a gun and you'd pull down parts off the shelf and you have to file each part to fit. And it was only by making parts with enough precision, famously in the Springfield Armory in Massachusetts, that people realised they could have an assembly line. As soon as you have enough precision that you can reliably fit different interchangeable parts, that's when you can make a factory and assembly line. And so then we move on to today, and now today we have the kind of MEMS devices that I'm sure you're all familiar with, where by using silicon planar technology, you're able to make electromechanical systems on the scale of tens of microns with precision that's very much better. And so the question then comes, where next? But first I want to just contrast, going from the left to the right. You know, at one level you think, oh, this is marvellous, how far we've come. You know, on the left we've got the dog for a scale, and now we've got a dust mite for scale. So on the one hand, you know, it's a huge distance, but on the other hand, it's still cogs and gears, and it's still a kind of recognisably mechanical engineering paradigm about it. So if you ask the question, where next? The answer that Drexler gave, the extrapolation of this trend, is very reasonable, but I think not right. Drexler's answer, of course, was that where next is that we just take the same idea of cogs and gears and we shrink it down to the level of individual atoms and molecules. And it's this idea of taking macroscopic engineering and shrinking it down that, of course, underlies all the depictions of nanotechnology in the media. So here's a kind of typical image that you see in the media of nanotechnology. So it's from a Microsoft encyclopedia, so it must be right. And you can see it's, so this is this vision of a nanobot in a bloodstream, cutting away the cholesterol deposits and sucking them up through little vacuum cleaners. Now, this image, it's a compelling image, but it's clearly, it's bonkers. It can't possibly be like that. And I think, you know, a good test of I mean, any scientific audience ought to look at that and say, this can't be right, it cannot be like that. But, you know, as so often is the case, it's actually worth digging down and scratching. Why couldn't this work? What's wrong with this? What's wrong with this picture? And the answer, of course, is this thing about scale. It's that physics doesn't work, it looks different on small scales to on big scales. So the kind of, you know, when we do engineering, we have a set of ideas about how physics work that inform the kind of rules of thumb and engineering rules that we use to do engineering design. And what's fascinating and different about nanotechnology is we're working in this different domain where a different set of rules apply. Well, I say a different set of rules. I mean, the laws of physics are kind of universal, but if you like, the approximations that are appropriate are different. So here's, you know, we've got something in water, and the way water behaves is different. So, you know, we want to make a submarine go through water. So we know that fluid mechanics tells you there's two reasons why water stops you going forward. You know, there's two possible reasons why water opposes you motion. One of them is inertia, that water's massive. If you want to go forward, you've got to give it some momentum. You have to exert a force on it, it exerts a force on you, it stops going. But there's another reason, this is this phenomenon of viscosity, that fluids have this kind of internal friction that when you try and move planes of fluid past one another, they dissipate energy. And so that's a quite separate physical reason why, to oppose motion. And fluid mechanics tells us there's a number called the Reynolds number, which tells us the ratio of the importance of those two things. So that's the formula. It's got density, a velocity, a size, and it's divided by a viscosity. It's the ratio of the inertial force to the inertial part of the force to the viscous part. And the key thing is it's got a size in. So at macroscopic scale, if you work out a Reynolds number for you going swimming, it's quite large. We're in the inertia-dominated regime. But if we shrink the size down by a factor of 1,000,000 to get us from our size down to a bacterial size, then you'd have to increase the viscosity by the same amount to get the same Reynolds number. So if we were to go, if we are shrunk down to the nanoscale size, you're in a completely different regime that's dominated by viscosity. And that has profound consequences. Low Reynolds number motion is utterly different from high Reynolds number motion. Most specifically, the laws of fluid mechanics, the Navier-Stokes equations in this low Reynolds number limit are actually time reversible. And most of the tricks that we use to swim in the macroscopic world would result in us going absolutely nowhere in the nanoscale world. Then we've got this phenomenon of Brownian motion. We know, you know, anytime you look at if you just look at a a nanoscale object, if we take and this is a colloid. If you look at a colloid, you've got particles being shaken around. We've got this constant shaking that comes from thermal motion, and that's got a size dependency in too. As you make things smaller, this thermal motion becomes more and more important. And it's not just a question, it's obvious that particles jiggle around. What's less obvious, though it's actually very well known to polymer physicists, is that Brownian motion affects the internal structure of things as well. So if you have a mechanism or a machine, all its internal parts will be jiggling. Equipartition tells us that every possible place you can put energy in will have a half a kT. So our kind of idea of machines as statically moving and engaging in a clean way has to be revised. We've got strong surface forces. The whole of colloid science is essentially the struggle to stop stuff sticking. When things get to be small, because van der Waals forces, it's a theorem that van der Waals forces between any two like objects are always attractive, and van der Waals forces are always additive. We think van der Waals forces are very weak. They are very weak in one sense, but they're additive. So when you have colloidal sized objects, they amount to really rather a lot. We've got things constantly stick, and of all the things in the world that are sticky, proteins are the most sticky of all. So any object that's going inside a bloodstream, for example, has to contend with the fact that proteins inevitably will stick with it. And we have this final question. We've got to make not just one, but trillions of these things to actually do anything. So at this point, we can just throw up our hands in horror in the one sense and say, well, this is doomed. We can't possibly make nanoscale machines. These obstacles that the physics of the nanoscale puts in our way clearly are too difficult to overcome. But of course, that's wrong. And the reason it's wrong, or the reason we know it's wrong, is because biology is utterly full of very sophisticated nanoscale machines. So here's this is just a video, a relatively realistic reconstruction of what a T4 bacteriophage does when it's affecting an E. coli. So this is a virus infecting a bacteria. And you can see this very sophisticated set of machinery that allows it to inject a piece of DNA into the cell. So it's wrong. Despite all these obstacles that the nanoscale physics puts in its way, it's clear that biology overcomes them, and biology does make sophisticated machines. So what lesson should we learn from this? Now, this is the point at which I think that, there's a right lesson and a wrong lesson. And I suppose to caricature the Drexler view of biology, and I think, to be fair to Drexler, if you read Engines of Creation, it's a very good book for emphasizing the wonders of biological things and, and taking that as a model. So, he was right, I think, to realize that biology has these great examples of nanoscale machines. And in fact, since then, we know much more about many more sophisticated machines. But he took the view, well, okay, biology shows you can make nanoscale machines, but, it's the best you can do with, poor materials. Engineers wouldn't think about making stuff out of floppy proteins, floppy polysaccharides, and these random design methods. So the corollary of that would be, as soon as you get a bunch of students on the job, people with nanotechnology degrees from top quality schools like Waterloo, as soon as these people get on the job, then we'll be able to make something much better. So by using kind of hard materials and mechanical design paradigms, you'll be able to make things that outperform biology. And of course, this is really where this idea of grey goo comes from. This idea of grey goo is that, we can make a machine that's a self-replicating device that takes stuff from the environment and uses that to make more copies of itself. What we're describing there is something like a bacteria. So what we're saying is, we'll be able to make bacteria that are better bacteria, that fill that niche of being a bacteria better than a bacteria does. What's wrong with this idea is this: biology isn't a thing that's hopelessly thrown together. If we look at the mechanisms of biological machines, and we can do this now with much more confidence than we could in the mid-80s, because single molecule biophysics, the tools of nanotechnology applied to cell biology, have allowed us to disentangle the operation of these machines in considerable detail. So taking this, for example, this is ATP synthase, a ubiquitous molecular machine Pretty much everything alive has got it. It converts energy from energy stored as a proton gradient into energy stored chemically in ATP. And the astonishing thing about it is that if you do the energy balance and you say how efficient is this at converting energy from one form to the other, it's very close to 100%. It's actually so close to 100%, it's actually quite difficult to measure the inefficiency. So, you know, if we compare that with the performance of energy conversion devices that we have in our macroscopic engineering, where, you know, you'd be feeling pretty pleased if a car engine was, you know, 30% efficiency. You'd be very pleased if a power station was operating at more than 50% efficiency. You know, these things really work very well. So the conclusion, I think, is that a better view is that biology is very highly optimized for this different physics at the nanoscale. And it's been perfected by billions of years of evolution. But so that tells us we should copy it, we should try and use soft materials, biological design paradigms. So on the one hand we should copy it, and the second corollary is actually we should probably stop worrying about grey goo, because we're really going to have a hard time doing better than biology. So why does it work? Well, I think if you look at biology and the way that cell biology works, it uses new design principles that are completely foreign to the world of macroscale engineering. And these are design principles that really follow from the different physics at the nanoscale. So rather than, you know, if we come to the nanoscale with an engineer's view and think these are problems that we have to engineer around, we'll have a particular mindset. But biology, not having a designer in that sense, just evolving good solutions, it exploits the different physics at the nanoscale. So the combination of surface forces and Brownian motion, for example, are brought together in this idea of self-assembly. The combination of Brownian motion and lack of stiffness gives us this idea of a conformational transition as a actuation mode. So the question is, can we just copy these design principles? Here's what I mean. Self-assembly is something that you get. Self-assembly needs two things. It needs a certain degree of programmed stickiness. So in that very simple example on the right, you've got magnets that are set up so that the green triangles only stick to the blue triangles. So that's a simple design rule that is encoded in the materials themselves. A much more complicated example, of course, is in a protein. A protein folds because there are various different interactions between the different amino acids. So the sequence encodes, you know, programmed stickiness, if you like. Now, it's not just programmed stickiness that you need. You need also Brownian motion. Brownian motion is crucial for self-assembly to work because the system's got to be shaken up. It's got to be able to try different configurations out, see which ones it likes, and then find the ones that work best. And so as you see on the right-hand side, eventually you actually see this non-trivial pattern arising. Now, biology does this in an incredibly sophisticated way. And this bacteriophage that I showed you before, this is a self-assembled object in the sense that if you take the proteins and nucleic acids that make that up, just put them individually into a test tube, give them a shake, they will form this fully functional structure that will do all that amazing machinery that will operate the amazing machinery that I showed before. So we can do self-assembly already, and the class example from soft matter physics of self-assembly is the block copolymer. And this is a very simple example because it's the programmed stickiness, if you like, only amounts to the fact that the red bit of the block copolymer doesn't like the blue bit. So you've got one kind of interaction strength, you've got only two parameters that control the geometry, the length of it and the ratio of the blocks. But even with that very small palette of things, you can still get very non-trivial structures. So the left-hand side shows you this remarkable long-range ordered structure called the IA3D gyroid phase, which is one of the nicest of the block copolymer phases. The right-hand side, that's a TEM showing you this very non-trivial nanostructure that spontaneously arises just because of the programmed interactions. You know, the structure, if you like, is encoded in that molecule when you make it. Why it's cool is because it's very simple, but it's completely scalable. If you can do this in the lab at the sort of 1 gram level, and it comes out nicely. But you can make this at the 10-ton level and it would still do it. And so, these things, they are commercially available. Some shoe soles essentially have structures that work because they're self-assembled like this. Here's another example from Sheffield. We can use these things to make bags. We can make molecular bags that we can use as delivery devices for molecules by the self-assembly, again, of these block copolymers. And you can see the micrograph there shows these very nice structures with wall sizes that can be controlled. Basically, you can dial up whatever wall size you like. This one's 2.4 nanometers, but you can control that just by controlling what molecule you make. The next idea is this idea of combining Brownian motion and lack of stiffness. If you ask yourself the question, how does a muscle work? There's a huge class of processes in biology that are driven by motor proteins. These are proteins that change shape in response to changes in their environment. And so in the simulation you see kinesin, which is making these hand over hands. It's moving. These things drag along materials in nerve cells, for example. But the point about it is that the power stroke, if you like, it's because the molecule is soft and squidgy that this works. And in fact, if we go back and ask the mechanistic question, this thing's changing its shape in response to things binding. But, from a Newtonian perspective, what actually is making the thing move? It's the Brownian motion. It's actually the fact that it's being collided with by surrounding water molecules that's driving this change of conformation. So there's a very elegant piece of physics due to Jacques Proulx in Paris. He produced this idea of the Brownian ratchet, this abstract notion that characterizes all these motors as being essentially rectifiers of Brownian motion. They're systems that rectify Brownian motion. Of course, that doesn't mean they break the second law, because you have to put energy in. They convert chemical energy, and that's how that works. So, why would you want to do this? Again, we appeal to scaling. So, if you're a proper engineer, of course, would want to have a heat engine. But we don't get heat engines in biology, and it's interesting to ask why. The reason is, of course, that a heat engine works by having a hot reservoir and a cold reservoir. It works because you have a temperature gradient, and heat flows from the hot reservoir to the cold reservoir, and you pinch a bit of heat out along the way, and you convert that to mechanical energy according to the Carnot limits. Now, as you make something smaller, the length of time that you can sustain a temperature gradient gets smaller, because big things get colder more slowly than small things. Small cups of coffee get colder more quickly than big cups of coffee. So the time it takes, the time that you can sustain the temperature gradient actually scales as the size squared, because it's a simple diffusion process. So as we make our heat engine smaller, we have to run it faster. Otherwise, the heat will have dissipated away from the explosion in the cylinder before we have a chance to get the mechanical work out. But of course, there's a limit. There's physical limits to how fast we can run the engine. Ultimately, the limit is, if you try and make the piston move up and down faster than the speed of sound in the material, it'll just vibrate, and you won't be able to extract useful work. So there's a crossover, and as you get smaller, these things just don't work. So biology doesn't actually use heat engines at all. It uses isothermal engines. It uses engines that are driven by chemical potential gradients, not by temperature gradients, and so should we. Here's an example, a very crude example of how we tried to make an isothermal engine to convert chemical energy into mechanical energy in our lab. It's a very simple responsive polymer that you can make by taking a polyelectrolyte. So this is a material, polymethacrylic acid, which is it's a weak polyacid. And so a weak polyacid has the characteristic that if it's in an acidic solution, it's unionized. If it's unionized, it's uncharged, it's hydrophobic, so in water it'll collapse to a little globule. If we then put it in a base, it'll ionize. Now it's a charged chain. The thing will be now hydrophilic, and it'll expand. So we can make a system that expands and contracts. Now, how do we clock the motor? Well, if we combine that with an oscillating chemical reaction, if we remember our chemistry, there's this very cool class of reactions, Belousov-Zhabotinsky reactions, the most famous, that spontaneously oscillate. They're essentially relaxation oscillators. You put fuel in the top steadily, take the exhaust out at the bottom, and they spontaneously cycle some quantity. We found a reaction that oscillated the pH over the right range to make these things work. So this shows an experiment in which we put our self-assembled polymer gel, which contains these responsive polymers, in an X-ray beam in this oscillating chemical reaction. So just to unpack this slightly busy composite video, the top left shows the macroscopic gel, which is expanding and contracting. Bottom left, this is the oscillating pH that this oscillating chemical reaction is generating. The bottom right is a cartoon of what's going on. We've got little self-assembled globules that are holding the responsive bits of the polymer together. And the X-ray scattering experiment, the scattering ring on the top right, is measuring the distance, the average distance between these globules. So it's actually measuring the average molecular size. And what you can see is that you get this, the thing is spontaneously throbbing, and you can see the molecular size, its reciprocal space, so when it's bigger the thing's getting smaller and vice versa. So you can see direct tracking of the molecular size to this chemical reaction. So that's our kind of very simple example of using responsiveness to do something interesting. So what should we do with this? Well, we could think about, we ought to learn nature's lessons. These are very powerful principles that we ought to use in nanotechnology. Two ways in which you could do it. Ned Seaman at New York University invented this great word, biocleptic, which I'm not going to take credit for, but it's such a lovely word, of stealing bits of biology. So the one approach is to actually pull out bits of biological motors and reassemble them in synthetic circumstances. So a very famous example is the demonstration by Montemagno of putting a bit of the ATP synthase motor on little posts and making propellers turn around. Alternatively, you could try and do nanotechnology in a biomimetic way, using the same principles, the design principles as biological nanotechnology, but using synthetic materials. Why do we want to do this? Well, I think people are more and more agreeing. What are the potential of of this kind of nanotechnology? I think everyone agrees that the kind of big challenges that we face, both societally and for economically, can be broken up into energy, getting energy into a sustainable basis. Information, organic-based electronics is something that may, well, it has arrived. The degree to which it becomes revolutionary is still not clear. And in medicine. I'm just going to return back to these societal issues, actually, because I think they're important. And one of the kind of textbooks, if you like, of the movement against technology is a book by an American journalist called Bill McKibben. He wrote a book called Enough, which is a very convincing book and very well written book, and in many ways very persuasive. And the argument in this book is that we have enough technology already. We've got these new powerful technologies, genetic modification, nanotechnology. It all seems very scary. We can see all kinds of things that could go wrong from these technologies. But on the other hand, we've got a comfortable life. Why don't we just not tempt fate, if you like, by stopping working on these new advanced technologies? And I think the important answer to this, and I think this is really important to make this point, is that one can say technology, technology's always been a double-edged sword, and it's always produced unforeseen consequences. But nonetheless, we have no choice. We have to use technology to solve our problems. And the reason for that is simply stated. If you look at the population of the Earth at the moment, the population of the Earth as it's going to be in the next 50 years, it is existentially dependent on technology in all kinds of ways. And one of the clearest ways that you can think about this is you ask, how much food could the Earth grow without the Haber-Bosch process? And one of your compatriots, Vaclav Smil at Manitoba, wrote a very nice book about the economics of agriculture, in which he very convincingly and quantitatively works out, even with everybody having a vegetarian diet and living off lentils, you'd be lucky to get more than about 40% of the world living off without the Haber-Bosch process. So we need technology, but on the other hand, we know what technology we've got is not sustainable. So we must get new technology to be able to sustain, to move over into a sustainable way of living. Energy is the most obvious example. Our demand for energy is continuously growing, and as the world's population it is going to stabilise sometime in the next century, but it's also become much more prosperous. We have this, historical process that we're living through at the moment of China and India getting living standards close to the ones that we're used to in Europe and North America, and that's a fantastically welcome development, but it's one that depends on a huge amount of energy. So we're going to need this energy. The only place that you can get energy sustainably is from sunlight, and everyone's got their favourite plot of the United States with whichever state they want blocked out about how much you'd need to pave over to meet the energy demands of the place. So that one's Western Canvas. Sometimes you see Nevada as well, but depends which one you want. The point is, of course, it's ridiculous to say you're going to put it all in one place. I mean, the whole point about solar energy is it can be distributed. So the question to ask is, well, why, if solar energy is so brilliant and there's so much of it, why don't we use it now? The answer, of course, is cost and scalability. The way that solar cells are made now is but using silicon by expensive, intrinsically high temperature batch processes. The products are excellent, they're efficient, long-lasting, but the manufacture is very difficult to scale. The total world output of solar cells is only of an order of gigawatt. It's only like one nuclear power station's worth of capacity arise every year. That's growing at 20 to 30%. It's very, very impressive, but the base is so low that it's inconceivable that unless something dramatic happens that this can solve the problems. What the dramatic thing to happen will be is moving to processes like this. One needs to move to processes that are very large scale. Now, many industrial processes that we have that produce stuff in very big areas, quite sophisticated things. You look at the humble chip packet, as I believe you call them, the South Atlantic. There's, they're quite sophisticated. It's polyethylene terephthalate base, metallized layer, probably some barrier layers to control the gas diffusion properties, probably one or two adhesion control layers, all of these things, sub-micron layers, that are produced in massive areas at virtually no cost. So this is what we need to do for solar energy. And of course there are ways in which nanotechnology can help, either the kind of bioeclectic version, where you can imagine harnessing the light harvesting complexes of plants and algae directly, or these more biomimetic ways that step back and say, what can we learn from the way biology does it? There's dye-sensitized cells, the so-called Grätzel cells, which have dyes on the outside of semiconductor nanoparticles. We've got organics photovoltaics using blends of semiconducting polymers and fullerenes, that sort of thing. Many different approaches that people are trying, but what they have in common is that they should be compatible with very large area, cheap processing. Now I want to finish off by coming back to this question of medicine, and I want to consider this question of drug delivery. Now, what is clear about when you take drugs, most of the drugs that you take don't actually do what you want them to do. This is actually a difficult measurement to do, but in some cases where people have been able to do this measurement, estimates are something like only 0.01% of an active drug molecule will actually get to where you want it to be. And okay, if you take an aspirin, that doesn't really matter because aspirin's cheap and it's not particularly bad for you. But it really matters a lot if you take, if you've got drugs that are highly toxic, so the kind of drugs that people use for cancer therapy, chemotherapy agents, very, very toxic molecules. We rely on the fact they're a little bit more toxic for the tumour than the rest of us, but it's not that much, and that's why side effects are so severe. Drug molecules are increasingly very expensive and quite delicate. More and more new candidate compounds are not simple small molecule organics like the traditional drugs. They're proteins, they're little chunks of proteins, bits of antibody. Delicate molecules that really are not easy to get into the body, and they're also heinously expensive. And I'm sure, in the UK, for example, there's been a huge furore about a drug called Herceptin, which is an antibody drug, very effective against some types of breast cancer, but very, very expensive. And the National Health Service really finds it, there's been lots of controversy about the National Health Service not thinking that it's cost effective because the stuff's so expensive. So these are serious issues, and they're growing more and more serious. So one way to get around it is to wrap up these drug molecules in some kind of enclosure. We can imagine this ideal drug delivery vehicle having an enclosure to wrap it up with, some coating to stop it being recognized by the body, perhaps some targeting agents. You can confuse things even more by putting in some kind of contrast enhancer so you can see where the drug's going through MRI or something. And all of this, of course, there are a few very crude examples of these things currently in clinical use. But all of this is starting to look like a nanobot, so it's getting us back to the nanobot again. And I suppose what we're saying is that actually this idea of a nanobot is not really such a dumb idea at all. It's actually what we're kind of coming towards thinking about these drug delivery issues. But of course, it's not going to be mechanical engineering shrunk down. It's going to end up looking much more like something biological. And we do see some early prototypes, things like so-called stealth liposomes, which is a technology used to deliver some anti-cancer drugs, where a liposome of the kind that I showed you earlier, a kind of bag that encloses the molecule, has a hairy coating on it to stop it being recognized by the body. And I want to leave you just in the last 5 minutes with this question of, okay, if we've got a nanobot, how can we propel it? How could we push it forward? And I remind you about all this issue about low Reynolds numbers. All those kinds of, having little propellers, little jet engines ain't going to work because we're in these low Reynolds numbers conditions. But my colleague at Sheffield, Ramin Golestanian, who's a very good theoretical physicist, had this very cunning idea for propelling one. He said the following. So supposing I take a particle, is my kind of model nanobot, and I half coat the particle with a catalyst. And I arrange the catalyst that catalyzes this reaction. I've got a reaction that has, doesn't really matter what the reaction is, as long as it does this, it produces more product than it has reactants. So I'm just sketching it in a cartoon as having something that's split in two, because that does the job. So it produces more products than reactants. But because it's spatially localized, we end up with a concentration gradient of those particles. And what this does is it makes a particle that generates its own osmotic pressure gradient. So it generates its own osmotic pressure gradient, which should push it along. And indeed, this video shows that that's exactly what they do. The three particles on the top, on the left and the top, they're control particles. So the top left has got no catalyst and no fuel. It just jiggles around doing Brownian motion. Top right's got fuel but no catalyst, so it just jiggles around with the classical Brownian motion. Bottom left has got catalyst but no fuel. So that's, again, just jiggles around. But you can see the bottom right, where we've got both the catalyst and the fuel, the little particle buzzes around like a little mad thing, really, and it gets a really significant velocity. And so we can analyse that in some detail, and I haven't got time to talk about that analysis, but it shows that this idea for propelling a particle really does work. And there's just the trace that just shows you the contrast between the simple Brownian motion of the unpropelled particles and the extended trajectory of the propelled particle. So, of course, other people think differently. This is our way of propelling a particle. We're clearly barking up a wrong tree. This is a depiction of a fertility treatment, a nanotechnology fertility treatment that involves slapping a rocket-propelled thing on the back of an underperforming sperm. So, with that, I think the future of nanotechnology, I think this idea of shrunken mechanical engineering, I think, it's interesting and fascinating, but I don't think it's going to happen because it neglects the fact that physics works differently on these small scales. And so probably we should stop worrying about, well, actually, I'm going to have to stop putting this slide up because the collapse of the money economy seems to be unwinding as we speak, but there we are, for different reasons. But, there are lots of very cool things that we can do with this kind of nanotechnology that learns from nature. I've talked about energy, medicine a little bit. The key thing, I think, this is slightly provocative because I think plenty of interesting nanotechnology isn't soft, wet and flexible, but I think there's going to be a great future in using these biomimetic ideas to do soft, wet, flexible nanotechnology. And I will close by, I can't resist mentioning, indeed, Jamie kindly mentioned this book, which is now out in paperback, which you will rush off and buy. I think we need to learn very strongly from biology when we do nanotechnology, as much from biology as we do from engineering. I want to thank the people at Sheffield. It should also be clear, and I don't have to tell you because you know this already, nanotechnology is intrinsically interdisciplinary. The work that we've been able to do has only come about because I've got a great collaboration with our chemists in Sheffield, particularly Tony Ryan, and our tolerant funders who, we wrote them a proposal saying we're going to make a nanobot swim, and they said, oh fine, here's the money. And I'm very pleased that they were tolerant of slightly speculative ideas that actually I think did actually bear some fruit. We did quite a lot of work at central facilities. I haven't talked about the data from neutron scattering facilities, but I showed you some scattering data that came from ESRF. We depend on those. Thanks very much for your attention.